Let's investigate a fascinating garden mystery today. In this explainer, we're diving deep into the science of plant diagnostics to uncover the stealthy, honestly devastating world of vascular wilts. It's a journey into forensic botany, and I promise you, it's going to completely change the way you look at your garden. Picture this for a second. You walk out into your yard, or maybe you're just checking on your favorite houseplant, and you see it drooping. Your very first instinct is probably to just grab the watering can, right? I mean, we all do it. But what if the problem isn't a lack of water at all? What if there's a silent killer lurking deep inside the plant itself? We've got a classic victim here, a plant suffering from a severe loss of rigidity. The leaves are dramatically drooping, literally looking completely exhausted. But here is the catch. Absolutely no amount of surface watering can cure this. Why? Because the issue isn't in the soil's moisture level. It's actually a total structural breakdown within the plant's internal biology. To solve this mystery, we're going to crack open the case file and methodically review the evidence. Our agenda today covers the scene of the crime, decoding symptoms, the prime suspects, hijacked plumbing, and finally, forensic diagnostics. Let's get right into it. Section 1. The scene of the crime, understanding how diseases take hold. First, we really need to understand how a plant disease even becomes a viable threat in the first place. Most infectious plant diseases don't just happen by chance, you know. For a pathogen to successfully attack, we need what plant pathologists call the disease triangle. 3 specific elements literally have to align perfectly: a vulnerable host plant, an active pathogen, and a favorable environment. If any single one of these sides is missing or weak, like, say, the environment just isn't quite human enough, or the host plant is naturally resistant, the disease simply cannot fully develop. But when all 3 align, that's when the balance tips and the disease strikes. Section 2. Decoding symptoms, identifying the true nature of the damage. To identify our culprit, we first have to correctly decode the forensic evidence left behind on the plant. So the absolutely crucial point here is understanding what true wilt actually means in a scientific context. Wilt is defined as a general drooping of the plant, or just a part of it, caused by the loss of turgor pressure within the plant tissue. Now, turgor pressure, that's just the internal water pressure that keeps plant cells plump and rigid. Think of a water balloon. When that pressure drops, you get a mechanical failure, and the plant essentially just deflates. But before we just jump to the conclusion that a hidden pathogen is at work, we have to let the symptoms lead our diagnosis. This means first ruling out abiotic or non-living stressors, like simple drought. Then we have to distinguish our wilts from other really common visible clues. We need to rule out blight, which is that rapid discoloration and death of twigs or foliage; canker, which leaves a dead, sunken area right on the bark; chlorosis, which is just a yellowing of the leaves; and necrosis, which basically means dead tissue. We are looking specifically for that catastrophic loss of turgor pressure. Section 3. The prime suspects. Introducing the vascular infiltrators. Let's see how our profile builds toward the true culprits behind these wilts. When professionals are trying to ID the culprit, they use diagnostic matrices to compare physical features. This helps narrow down whether the attacker is a fungal, bacterial, or viral pathogen. For example, fungal pathogens often leave these circular or irregular lesions that expand gradually. Bacterial pathogens, on the other hand, might show water-soaked, angular borders and spread super rapidly in humidity, sometimes even producing a visible, kind of gross ooze. Viral pathogens often present as a systemic model or a mosaic pattern on new growth. Comparing these features is an absolute game-changer for narrowing down our suspect list. For our specific botanical mystery today, our main suspects are the devastating fungal culprits, Verticillium and Fusium. These are sneaky, soil-borne fungi that infect hundreds of different host plants. And get this, they don't attack from the air, they infiltrate from below, breaking in right through the root system. Verticillium alone has over 200 host plants. It attacks everything from massive woody trees like maple and chestnut, right down to the tomatoes and peppers in your backyard garden. Section 4. Hijacked plumbing. How the attackers operate internally. Once these fungal suspects break into the root system, how exactly do they execute their crime? Let's take a look inside. They target the plant's internal plumbing. A healthy plant relies on 2 main vascular systems. The phloem conducts sugars and organic nutrients outward from the leaves to the rest of the plant. But our culprits, they go straight after the xylem. The xylem is the absolutely crucial system responsible for pumping water and vital minerals upward from the roots. When the xylem is attacked, this tragic biological battle takes place right inside the tissue. The plant realizes it's under attack, right? So it desperately attempts to defend itself by sealing off the infection to stop it from spreading. But here is the heartbreaking part. By intentionally blocking those vessels, the plant inadvertently clogs its own water supply. It essentially strangles its own plumbing system, trying to save itself. Section 5. Forensic Diagnostics, Revealing the Hidden Evidence. Now, we deploy forensic botany to prove definitively that the xylem has been hijacked. This is where we find definitive physical evidence. Whether it's Fusarium wilt in sumac or a verticillium infection in a maple tree, if you cut a cross-section of the stem near the base of an infected plant, you completely expose the crime scene. You'll actually see distinct brown and black streaks forming rings of vascular discoloration. In dicots, like those trees or a tomato plant, it forms this dark ring right around the central pith. That discoloration is the smoking gun, visual proof that the xylem has been colonized and totally destroyed. In a professional lab setting, diagnostic specialists take this a step further. They use specialized chemical stains to reveal these invisible blockages under a microscope. By applying a dye called safranin, the lignified walls of the xylem are stained a bright, vibrant red. Then they use fast green, which stains the phloem green. This high-contrast analysis allows scientists to perfectly illuminate the vascular bundles and physically confirm the total collapse of those microscopic water pipes. When you get a clean cross-section, that dark, discolored ring represents irreversible vascular clogging. Because these pipes are permanently blocked, both by the fungus and the plant's own defensive barricades, water simply cannot travel up the stem anymore. And this is exactly why watering a plant suffering from severe vascular wilt is completely useless. The water is sitting right there in the soil, but the internal highway is permanently shut down. This brings us to a pretty harsh reality, perfectly summarized by guidelines from the Plant Disease and Insect Clinic. They state, if your plants have bacterial wilt, there is nothing you can do to save them. Infected plants will not recover and should be removed and destroyed. Now, the exact same devastating rule applies to severe fungal vascular wilts. Once that internal vascular system is heavily compromised and discolored, the victims simply cannot be saved. The absolute best course of action is safe removal to protect the rest of your garden from the spreading pathogen. Which brings us to a final thought for this explainer. Before you instantly reach for that watering can to cure a sad-looking plant, take a closer look. Next time a plant droops, ask yourself, is this plant really just thirsty, or is there a full-blown botanical crime scene, a true vascular wilt, hiding just beneath the bark? What forensic secrets will you look for?